Hello, everyone, and welcome to SOI's first Back Ashore of 2023, a series where we revisit with scientists and engineers that have sailed on Falkor and learn what has happened since they have come on board to complete their at-sea expedition. My name is Allison Miller. I'm the Senior Manager of SOI's research portfolio, and I am here with Dr. João Sousa, who sailed on board in 2018 from the University of Porto, Dr. Blair Thornton from the University of Southampton, who also sailed on board Falkor in 2018, Dr. David Gruber from SUNY Baruch College, who sailed on board Falkor in 2019 and also developed some technology that we brought on board and integrated onto ROV Sebastian in 2017. Dr. Kakani Katija from Monterey Barium Aquarium Research Institute, who sailed on board in 2019 and on the second to last expedition of research vessel Falkor in 2021, and Dr. John Burns from Bigelow Laboratory for Ocean Sciences, who was also on board on that same expedition in 2021. I'm sure all of you know that the science and technology that we see live streamed and hear about taking place on Falkor is really only a small glimpse of the development, analysis, building, and instrumenting all of the technology that comes on board Falkor. So I'm really excited to hear from all of our panelists about what they've been up to since they walked off of the vessel and how their technology has evolved and what they've learned using their technology at sea. So all of our panelists can describe their technology and do it way more justice than I could. So I'm going to start by letting each of them describe the technology they used on board Falkor. Let's just start in the order of introductions, and we'll start with Dr. João Sousa. Could you tell us about the technology that you brought on board in 2018 and more about your project?
Yes, definitely. It's a pleasure to be back. Thanks. So we developed an integrated multi-vehicle system capable of finding, tracking, and sampling dynamic features of the ocean. Our target area was the Northern Pacific subtropical front. And to do that, we used 7 AUVs, 2 Sailtrons, 1 wave glider, 2 VTOLs, and other quadcopters. And with the help of our software, we were able to coordinate their motions so as to sample a segment of the front in the best possible way.
Awesome. I'm really excited to hear from you and hear what you've been working on since that was almost 5 years ago now. So it's really great to see you also. And Dr. Blair Thornton, you were also on board in 2018 doing some work with multiple vehicles. Could you tell us what you were doing on board?
Yeah, so we were an international team of marine robotics engineers from the UK and Japan, and we had multiple AUVs, not quite as many as Joa had, but we had 3 AUVs and also the ROV Sebastian. And these AUVs, they were equipped with cameras and chemical sensors, and we were trying to map multi-hectare regions of the sea floor this time at sub-centimeter resolution. So although we had multiple AUVs and sensors, the actual technology we were developing and testing on the Falkor was a series of algorithms that we were using to rapidly summarize the enormous volumes of data these systems were gathering. So instead of kind of doing a lot of processing when we were back on shore and trying to understand what the robots had seen, we would get a quick summary of what the robots had seen kind of between the cycle of dives so that we could have kind of better informed plans on what to do the next day when we're retasking the platforms.
Excellent, thank you. And David, you developed some technology that came on board without you in 2017. Could you tell us a little bit about that technology development project and then also your role when you came on board in 2019 and the technology you brought with you then?
Sure. Yeah, these were squishy robot fingers. And a lot of this technology was inspired by being in a submarine. I'm a marine biologist, and seeing animals at the bottom of the ocean that looked very new to science, but wanting to interact with them in a way that didn't hurt them. So a lot of the kind of titanium claws are not really meant to be picking up really delicate animals like jellyfish and siphonophores. So this was the beginning of an iteration of numerous technologies, including a rotary actuated dodecahedron that was used on this cruise and combined with Kakani's scanning work, and as well as John Burns, who's on the call, really leading the way in doing the genomic work. So working together and collaboratively, we're aiming to create what we call digital holotypes, ways we could study creatures at the bottom of the ocean without bringing them to the surface.
Excellent. And Kakani, David teed you up well, but could you talk a little bit more about the technology that you use?
Yeah, so our first cruise out, 2019, we brought DeepPIV, which is an imaging system that combines laser illumination to scan animals, particularly gelatinous animals, to do complete 3D reconstructions of their bodies non-invasively. And then when we went back out, so that's DeepPIV, currently scanning a siphonophore. And then when we went back out in 2021, I think that was the year, not only did we bring DeepPIV, but we also brought a new imaging system. It's a light field imaging system called IRIS. And that's what you see here. It's equipped with, I believe, up to six lights that are on an expandable array. And then there's a single camera within the housing there. And we're currently doing measurements on a siphonophore as well. And so what we're able to do with IRIS is get, fully volumetric data in real time, but also time varying, so at 60 frames per second. So that means we can do things like 3D measurements, reconstructing of surfaces, particle and animal visualizations, etc.
Awesome. And John, you were on board the same expedition. Is there anything you'd like to add to what Kakani said?
Yeah, so I'm a molecular biologist, and my role was to help try to understand the genetics of these animals because they're so new and we don't really understand how they work. And I got first involved with this maybe back in, I think, 2019, 2020, when we were doing kind of stress tests on jellyfish, and we were wondering, like, if we want to understand how these things are existing underwater without really freaking them out, like what kind of technologies and what kind of protocols do we need to employ to do that, so that we get a sense of how they live in their natural environment, rather than what they look like once we bring them up here and take them way out of what they're how they exist in nature.
Great. Well, you teed up my next question really well, but I was going to take a step back, and before we talk about how your technology and the projects that you conducted on Falkor have advanced or moved forward since you've stepped off the vessel, I wanted to know what your development process was like, and really how you conceptualized your project. Were you trying to solve a problem? How many iterations did it take before you really felt like you were ready to come out to sea and test your technologies? And John, you alluded to this a little bit with wondering about what the jellyfish were feeling. So maybe I'll just ask you again if there's anything you wanted to add now that I've asked the question to what you just said, and we'll go around the table from there.
Sure. So those stress tests were with David Groover and who's Mercer Brugler, and they were testing like a jellyfish if you grabbed it with their gentle squishy fingers, or if you grabbed it with a hard claw. And then we went and looked at the genetics and saw that when you grab it with a hard claw, it gets more stressed out than if you grab it very gently. And I think that was part of the motivation to try to grab it with something like this flower, the rotary actuator, the RAD device there, that kind of just encases it without even touching it, right? So the goal was to really get this information from the animal before you had a chance to like really freak it out.
Excellent. David, would you like to add anything to that?
Yeah, I just to follow up with that, it's such it was a really fascinating question to us of like, you know, as we're designing the technology and we're aiming where our intention is to be really gentle, but it begs the question, like, how do you ask a jellyfish if the technology you're working on is not stressful to the jellyfish? So we thought long and hard about that. And we looked at, well, we could look at the jellyfish stress genes. But it wasn't very well mapped. When you look at some of these marine creatures that you find at the bottom of the ocean, you might find like barely any genetic information just because they're so hard to get, or humans have just really rarely interacted with them. So we don't have much like blueprints to look at. Like if we're working on a human, there's this massive amount of human information that we could work with. But like the siphonophore that Kakani was showing, there might sometimes there's zero. Sometimes this is an animal that's completely new to science. So the technique that John was talking about is that we looked at every single gene that was being expressed. And first we just looked at a resting jellyfish, and then we analyzed it with using the squishy fingers. And we were able to see that the squishy fingers didn't stress it. So that's how we got to the bottom of the mystery of, are we stressing out the jellyfish?
That's amazing. And then along those same lines, Kakani, you kind of took that a step forward and started using 3D imaging in situ to hopefully create even less stress around the organism. Would you like to talk a little bit about how that technology was developed and came about?
Yeah. So DPIV, by the time we came on the Falkor the first time, we had gone through several iterations of that instrument. So I think we were on like version 4, and it had been we've been diving it since 2015 actually, and being able to really dial in how we use it, how we operate it at Mbari. But this was actually our first opportunity to deploy this instrument off of someone else's ROVs. And so that was a really important test for us, right, to make sure that the technology we've been developing can go elsewhere instead of only here at Mbari. And then when we brought IRIS on board, it was actually a much newer instrument. It was, I think, only within the first 2 years of us deploying it. And really one of the first opportunities to answer or try to help address this problem of, how do you create a description of an animal as quickly as possible in the ocean, right? So that you can perhaps help with describing new species as quickly as possible. And so, in this case here, we have IRIS, and not only are we with DPIV creating 3-dimensional scans and models that can be used for, interrogating animals after the fact. But then with this instrument here, we're able to do really fine scale, like sub-millimeter measurements on specific features that each of these animals might have. And so this information is, of course, really important with morphometrics, understanding different morphologies that animals have. And so being able to combine all of these things all together, right? The genetics tools that John and Dave and Brennan were developing or creating, the collection mechanisms as well as the imaging systems all together, I think, are really powerful approaches. And so this is, an image of one of the IRIS outputs where in this case, this is what we call a total focus image. So we've collected so much data, but we've already been able to put 3D models of these animals we were studying from this cruise online and available to everyone.
Yeah, seeing those models has been really amazing, and I'm excited to hear more about them. Blair and Joao, you had completely different themes for your expeditions and focused more on the robotic aspect of exploration. Could you talk a little bit about what inspired you and brought you to Falkor and how your technology developed before you came on board, or what you're trying to solve when you came out? Blair, I'll let you go first this time.
Okay, yeah. So in our case, it was probably about the 50th expedition where we've been deploying AUVs with cameras. So we probably deployed camera-equipped AUVs maybe 200 or 300 times before we came on the Falkor. And what we were basically doing is we're trying to deal with our own frustrations that we had faced when we were doing this work. So during a dive, an AUV will take maybe 10,000 photos of the seafloor in a couple of hours. So in a few days, you end up with more photos than most people have stored, on some online photo server that automatically sucks up all the photos from your phone. So just imagine scrolling through your entire life's digital photo album, how long that takes. So that's the kind of exact same situation we face between the dives of an AUV. We want to understand what it had seen, but we're unable to because it just takes us too long to kind of scroll through and try and understand what the AUV had seen. And if we're out at sea for weeks and data's coming in every day, we very quickly start to fall behind with what's going on. And then basically after a few days, you have no idea what data was being collected. So what we have been doing was, like in the image over here, we were developing technology to build these kind of 3D reconstructions of these large areas. So there's a football or a soccer field drawn roughly to scale over there, but we're imaging quite large areas and we're building these three-dimensional reconstructions. We're also developing technology to summarize the data that we had gathered. So we had algorithms that would try and group images into similar categories. And instead of us having to look through tens of thousands of images, the algorithms would pick just a handful of the most representative images in the data set and kind of the distribution of where similar looking things to that were in georeference coordinates. So that would kind of help us understand very quickly and build up a good picture of what the vehicles were seeing. And we're able to do that in hours. So next time we deploy the vehicle, we can have much more targeted and focused plans. Yeah, so this is an example here. The bottom left shows a colorful map. So that maps onto the previous photo mosaic. So the thing about the photo mosaic is if you zoom in and scroll around, you can start to build up your understanding, but that takes some time and it's very difficult to share. Now, what this shows is a colorful version of that. And on the top right-hand side, what you're seeing there is a tree map. So that's 60,000 photos arranged and grouped in order of how similar they look to each other. The thing below it is a selection of 6 of the most representative images of each of those groupings. So if you kind of look at the map on the left, you can see that there's like a blue ring, and in the center of that, there's a green patch, and then there's some pink blobs and red lines. And if you look on the bottom right at the most representative images, we very quickly start to understand, okay, so the blue rings, that's sand and sediments. And in the middle of that, we have rocky outcrops. The pink patches are these bacterial mats, and then the dark red regions are these bits of cable and infrastructure on the seafloor. You don't need to scroll through tons of images or tons of data to do that. We have these automatically generated summaries that help us do our job better when we're out at sea.
Excellent. And Joao, I think you are also trying to solve a big data problem, but more identifying physical oceanographic targets rather than biology. Could you talk about how your technology came about and was iterated upon?
That is correct. So we went after the Northern Pacific subtropical front, which is a place where kind of Arctic waters meet waters that are somewhat warmer. And there's a very strong salinity signature, but a very weak temperature signature, meaning it's very difficult to see from space using SST And at the same time, it's mostly quality. So one of the issues was to find the front. So we did that with one wave glider in 2 sailtrons. We were measuring temperature and conductivity in real time and trying to find the signature of the front. And it worked well. So, and in terms of technology, we built on our experience with coordinating multiple assets, underwater, surface, and air, and expanded it to address the problems that we would be facing in this cruise. So first of all, software. So our software was evolved to support multiple control centers, one on board the Falkor, the other one in Portugal. This was embedded in the cloud. Then we were communicating with the assets, with most of the assets using Iridium SATCOMs. And then each asset also had its own software that had some intelligence to sample the front. So at the same time, we were also able to get data from the field in close real time. So we could, in close real time, reconstruct the observations, in this case, in terms of salinity, in terms of temperature and other variables. And based on those observations, we could redirect the sampling efforts. And we did this for 2 weeks, operating nonstop in shifts. And the interesting thing is that although we are operating a huge number of assets, we only had 2 guys operating these assets. So we are operating in shifts and 2 guys per shift, except when we are running air operations like this one with these VTOL. Something that we did just to minimize cost and to be good to our hearts was, so we developed 7 new AUVs that were supposed to be low cost, and actually these were low cost. In terms of endurance, these could go up to 60 hours, and these so we were quite relaxed about the operation of these guys. Why? Because if something went wrong, it wouldn't be a major disaster. And in the end, everything really worked perfectly well, and we were quite happy with what we achieved. There's also something that we achieved that has nothing to do, let's say, with technology, but simply with the fact that on board, so we had physical oceanographers, chemical oceanographers, marine biologists, engineers, people from informatics. And initially, we were not exactly working as a team, but a few days into the cruise, I'm pretty sure that if you went to some of our meetings, you couldn't tell, hey, this guy is an engineer, this guy is a physical oceanographer. So this is something, this is a kind of interdisciplinary work and planning and execution that I think was also a very important achievement of our cruise.
Yeah, thank you for bringing that up. I think integrating the technology developers with the scientists who are analyzing the data and creating impact with that data, it's really important. So that's a great point. I also forgot to remind our audience to type in any questions they have for our panelists. So please feel free to do that and we'll get to those. Everyone mentioned great science and technology that took place on board Falkor. And I wanted to know, what was the first thing that you were really excited about when you stepped off? So what did you learn on board the vessel during your expedition that you were ready to walk off and get back in the lab and either reiterate your technology, or you had new questions that you were excited to use your technology to answer, or just did any parts of your mission change, maybe mid-cruise or once you got off? Kind of what were the next steps when you got back to the lab? And I'm just going to leave it open. So if anyone wants to answer that, please feel free. I stumped you already, and we're only on the third question.
I think we're all just being too polite. I think you'll have to choose one of us.
Okay, David, go ahead. You're already unmuted.
I mean, first off, for us scientists, I mean, we can't understate the opportunity to use the vessel like the Falkor and how much that advances our science. You know, it's kind of like our dream is to be getting on board vessels like this to be able to do the experiments that we're describing. You know, so a lot of the time we're writing grants, we're trying, you know, we're doing the science, we're trying to, you know, position ourselves. So it's kind of like, you know, I think all of us would say that it's like being, you know, a kid in the candy store. And we have access to this amazing boat and all the tools and resources that it provides. And even stepping back for a second and thinking about some, you know, how difficult it is to get down 4,000 meters under the ocean is not something every boat has the capacity to do. So, you know, even asking these questions of how would we look at jellyfish, you know, deep in the deep sea, gets us excited how we could study these things. So yeah, I'll just kind of frame it from there and then, you know, in a broad thing, but glad to go deeper.
Thanks, David. And Connie and John. Oh, yeah, please go ahead, Joao. Oh, you're muted. No, please go ahead.
I would just like to allude to something that I think it's very important, and we learned something about it during the cruise. Now it's obvious, but it was not obvious at the time. So the op area was quite large. So initially we thought, hey, we found the front, so we put AUVs in the water, and the data that we were collecting suggested, hey, we have Arctic waters here and warmer waters here. But while we were doing that with AUVs, which are not particularly kind of fast, we went south with the Falkor, much faster. And then what we saw was somewhat puzzling, because we went from, let's say, cold waters to warm waters, cold waters and warm waters again. This was a sort of not what we were expecting. And the only reason why this was very useful, so different speeds and being able to select different areas and study those in parallel, was that then we concluded, hey, we really don't understand what is going on here. So we decided to go for a large, kind of a sort of a mesoscale mapping in the sense of size. So something like 50 nautical miles by 40 nautical miles, had AUV sampling all this area. And it was only after that we understood that there's a front, and then there was a filament detaching from the front. And let's say, I just want to be as succinct as possible. I think that something that we need in the future is to be able to combine speed to get to some regions and to sample as fast as we can, so that we learn about what is going on there, what is going on in other regions or so, to get a sort of a global picture of the area, so that after that, then you can start planning in detail what you want to do. So in this combination of speed and the capability of deploying multiple assets that are collecting data concurrently, I think it's very important, and I think that we can do much more with that.
Yeah, maybe sending out some autonomous vehicles before the ship would help with that speed. Yeah. Does anyone have anything to add to that question, or I'm happy to ask another one, but go ahead, Kakani.
I do. A few things. I would say one of the first feelings I had when we got off the boat was relief that our systems, right, worked great. Not only, did they work, like being able to interact with and work with ROV pilots who are fantastic, and made me realize, right, this is something that can be exportable, that other groups can use and collect information and data. So that was really important for me, right? As an engineer, I want to make sure the stuff that we spend a ton of time developing can go elsewhere. But then the second thing, which funnily enough, I was not at all expecting this, but I was watching the live stream feeds during our dives and noticing the same people showing up on the YouTube channel, commenting, having discussions with each other, excitement around all the stuff that we were doing. And we started talking with them through this YouTube, chat, and started to find out that there's this immense community of people. You could go on the live stream oceanographic Discord server, and these are very, very dedicated enthusiasts that watch all of the ocean live streams. And what it did is it kind of opened up my eyes to the enthusiast community that is out there, and has made me realize that there's this big group of people that want to participate in ocean science more directly, and be able to move the needle, let's say, in ocean exploration discovery. And so that actually, with that information, started thinking about how we could potentially do that. So it's yes, we're still doing a lot of this imaging systems, instrumentation, robotics stuff, but we've started to transition to think about how do you create engagement through data, through imaging, and interact with groups like Livestream Oceanographic.
That's an excellent point, Kakani. And I actually wanted to ask, how do you guys feel the best way to communicate about the new technology that you're using for oceanographic research to the general public and also within the community? Typically an academic publication, I know there's robotics and technology focused journals, but typically publications are more reserved for scientific outcomes and not technology. So how can we best communicate what technology you have developed that is available for others to use, and also, Kakani, like you mentioned, to the broader public? Does anyone have any input, or should I pick on people again? Okay, I'm gonna pick on Blair this time.
Yeah, well, I guess the ocean, especially when we do things, I'm a professor at a university, so we do things like public outreach, and we talk to all sorts of different age groups. We talk to people who have a technical background, people who are just, visiting because they live locally in the area. So I guess one of the things is that I've noticed is that everyone has kind of a slightly different perception of what the ocean must be like. Some people think, will assume that it's cold, there's nothing down there. Other people will be just naturally intrigued by it. Other people will think of it as a frontier, like, outer space. So actually it's interesting when you talk to people, because they all have different starting points. And when you try to explain your technology to them, if you don't take that into account, you're talking different languages, and it's not this kind of interactive thing. It's just a blurb. So I think that's something that's quite valuable, is having that two-way interaction and being able to understand, okay, so this is this person's perspective. How can I make what I do relevant to their interests and their perspectives. So I think that's something that's really incredibly valuable, but it's a bit of a skill set that we're not necessarily trained in through academic peer review processes or grant writing processes. So it does take, putting yourself out there and your technology out there, and almost kind of, exposing yourself to all of these kind of different perspectives to actually understand how what you do kind of fits into the bigger picture of things.
Definitely. John, I see you nodding your head. Do you have anything to add?
Oh, I was thinking about, so while he was talking about outreach, I actually, this particular cruise, the Falkor cruise that I was on, someone asked me to give a presentation to a 4th and 5th grade class in Canada. And they were like thrilled, you know, and the vampire squid behind Kakani and David, they loved it. Like, you know, like, so it's such a bizarre and alive environment that we never ever get to see. And when you bring it to life with the images and even the genetic information that we're getting, it seems a little bit abstract, but some of the things that we were surprised by was like a couple of the siphonophore, this tomopterid, a couple of the siphonophores have a genome, so that's all of their DNA. That's more 10 times the size of the human genome or more maybe. And we didn't expect that at all. Like why do they have so much DNA in them? What is it all doing? And when you can relate it that way, like to these bizarre animals in this bizarre environment, they're beautiful. It can really like spur people's imagination, especially 4th and 5th graders. But I think, you know, it works pretty broadly. And with the way that cruise was structured, we're working on a scientific publication, but it's an integrated one that integrates technology, genetics, and kind of broader questions about how do we want to conduct ocean research, at least ocean biological research.
Great. Yeah, that would be good. David, when you developed squishy fingers, I don't want to say they went viral, but they became pretty popular, I feel like, in publications and the media. Do you think there was anything specific that you did really well with that technology, or you were just using them to capture charismatic fauna and people grasped towards that?
Yeah, I mean, calling them squishy robot fingers kind of helped, I think. You know,
are we allowed to call them that, or should we use something more scientific?
No, you're totally allowed to call that. Yeah, yeah. No, I mean, yeah, I mean, sometimes, you know, we just have to step away and, you know, realize that sometimes we think our work is very serious, but also take the fun side of how cool it is that we have these jobs and these opportunities to be thinking about that. One of the other, I think, reasons why squishy robot fingers really stuck was the beginning is that this was, well, I'm a National Geographic explorer, so was Kakani. They have just tremendous outreach, you know, and storytelling, and really thinking a lot about storytelling. I think now the narrative blends itself into the research. Like, I really think about the storytelling aspects from the very beginning of the research, and that might even pick what areas we're doing. For this one, I think, you know, and John mentioned we're writing a collaborative paper that we're spending a lot of time, you know, working on it together. And then after that comes out, we're excited to like share that with the world. There's already some media that's expressing interest in it. There was a piece just recently called The Challenge of Deep Sea Taxonomy, you know, just posing this question of like, how will we humans study the deep sea in the future? You know, we see technology moving so fast, and I just like it. You know, it kind of allows us to think in this futuristic mindset of how are the kids that are watching this now, you know, how will they be doing marine biology, you know, and when they're grown up.
That's an excellent question, and let us know when the paper comes out. We'll be happy to share it as well. We do have some audience questions, but before we get to those, I wanted to give each of you the chance to let us know how your technology that you brought on board Falkor 2 to 5 years ago, how has that changed or advanced? Have you used it since on other expeditions? Are you back in the lab? Building upon that, we would just love to hear what you've been up to. So maybe we'll go back to Joao, who was on the cruise lease recently.
Okay, so it really helped a lot because let's say our core business, if I can say that, has to do with coordination of multiple assets and coordination to achieve some goal. The goal has to do with sampling or something else. And so we learn a lot from the cruise, and we've been expanding what we use there and what we developed there in other exercises. And I would just like to allude to a large exercise that we've been organizing in cooperation with the Portuguese Navy and NATO. Surprisingly, it's not a military exercise. It's evaluation and testing exercise in which we evaluate and test different vehicles in an operational scenario. And it's large scale, and by large scale, I mean last year we had over 120 autonomous vehicles, so underwater, surface, and air, and close to 20 ships. And this poses lots of questions. Okay, how do you coordinate? What are the issues? You have different vehicles. These vehicles don't talk to each other or so. So how do you address that? And we started addressing that with some of the tools that we used in Falkor. For example, instead of having one control center, because we had multiple ships, having one control center in each ship, and then having those communicating for situation awareness. So I think that definitely that is very important, and this type of exercise is also quite relevant. And I should also mention that not only it's not a military exercise, but we bring together the so-called triple helix, so academia, industry, and government armed forces. And everybody works together to achieve goals that are of interest to most of us. So definitely we learn a lot from our cruise, and that has been transitioned, for example, to some of the experiments that we've done in this exercise. And the idea is to keep growing in scope. And actually, I would also like to allude to this book by Ray Kurzweil and he talks about exponential growth. And actually, in this discussion, I think everybody mentioned one way or another way that we are going through a sort of a different era, if you will, in which you have more assets, more systems, and I believe that to a certain extent we are witnessing some exponential growth in some tools and technologies. And by doing that, by playing with these vehicles or so, then you learn how to handle many more vehicles in the future.
Yeah, great point. I'm glad that your project also opened you up to new partnerships around the world and in Portugal. Blair, could you share how your project has advanced and what you've been working on in the past 5 years?
Yeah, sure. And but I also just wanted to kind of pick up one point just on the earlier thing about kind of communication and stuff. The things that I've noticed is when we kind of talk about the technology that we've developed and all those things, a lot of the time people are also interested in kind of the human aspect, you know, how was that technology developed? So when you talk to people in the public, sometimes you're telling them about, you know, what you did on this cruise, and the questions are, you know, what's it like to be out at sea for so long? You know, what do you eat? You know, do you have showers? And there's those kinds of questions. And, you know, rather than pushing back on that and saying, no, you know, let me tell you about the camera. It's good to kind of engage with that side of things as well. So I just thought it was a good point on the communication, is it's maybe there's the story behind the technology, which is relatable to people. But yeah, sorry, that was a diversion. But in terms of what in terms of what we've been doing since, so we've been, you know, continuing plugging away at this kind of rapid summarizing of data and information. I think it's incredibly important for us to manage how we work, but also I think it's that there's a lot of parallels that we're noticing. It's not just to do with imagery or the kind of seafloor mapping data that we gather. Kind of the ability to pick out what's important, what's surprising, and what's representative from data. It seems to be something that's just generically quite important as sensors become have higher and higher resolution. They measure over wider areas. The vehicles travel faster, further. It becomes more and more important, because the gap between what a human can process and understand from the data and what the machines can go and just gather automatically, starts to get bigger and bigger. So we need kind of a helping hand to channel that through to something that we can kind of understand quickly and make decisions based on. And one of the things that we've been doing recently is we recently completed a ship-free shore-launched AUV operation. So this was using the boat face. May have heard of it, but it's a long endurance AUV. It can operate, well, in this particular instance, it operated for 20 days, and we did a cruise that was over 1,000 kilometers long with no ship in the water. So the ability to kind of filter and narrow down data is very important, because when you've got a robot out there on its own, it can't send the data it's gathered over a satellite antenna. One of the photos that we take would take about 5 hours to send just one of those photos, raw through a satellite. If we take 100,000, we're talking decades to send that data across. So that ability to summarize is something that's kind of enabled us to retask and manage robots when actually we don't have humans on boats out in the field. When the robot is out in the sea on its own doing one of these long-term missions, and we're in our offices, I'd be sitting where I'm sitting now, able to kind of understand what the robot's doing and retask it. So I think that's the biggest development that we've taken. The biggest direction that we've taken things is actually kind of diversifying the applications of the technology that we first demonstrated on the Falkor.
That's awesome. So great to hear. David, could you talk about how your technology has advanced since 2017 and 2019?
How has our technology advanced? Well, I mean, that's the fun thing about science. It's constant trying and redoing. So, in thinking about squishy robot fingers in the earliest days, we didn't even know if soft robots would work well underwater. So we just tried, even bringing them down to the deep sea was our first questions. So we went from squishy robot fingers to squishy robot wrists to squishy robot arms to a glove that could move a squishy robot arm to the rotary actuated dodecahedron that you see encasing the jellyfish, to now this kind of collection mechanism that allowed us to do this genetics. So I think the next steps are actually to continue to bring these things together and think of, we were so excited to bring in Kakani's work into this and develop this kind of workflow. So in, I think about 20, 30 minutes, we could first scan, and then it gets, and then it goes into the rotary actuated dodecahedron, and then it gets sampled. What we're thinking about is like, how can we make this even more gentle? Is it possible that we could use like a squishy robot finger to just rub on the side of one of these animals to get the cells off and then do the analysis from there? That would be a next step, but it's always like stepwise. We're going one step at a time to get more and more delicate, but we're understanding that this is really hard. We're working from a remotely operated vehicle in the deep sea. So we just take it one step at a time. So we're excited to the next step would be to use like medical biopsy and be as gentle as like a doctor doing a checkup in a doctor's office. That's our next goals.
Yeah, constantly moving that finish line forward. So I love that. And what you guys have achieved so far has been amazing. Kakani, Would you like to share what you have been up to?
Just a few things.
Nothing much.
Well, I mean, so for instance, with Iris, this is one of our first forays into light field imaging or multi-view imaging. And it's a very, very powerful technique with lots of different applications, lots of different ways you can orient or, put your cameras and illumination to get different fields of view, different resolutions. So we've been thinking a lot about scaling up some of the imaging systems so that we can interrogate larger bodies, larger systems, et cetera. We've also taken Iris on a number of other expeditions, including one to Davidson Seamount last December. And there is an amazing dive site. It's called Octopus Garden. And I think that was only discovered a few years ago. And we were able to get the first measurements, or at least some of the first measurements of biomechanics of crawling and swimming octopuses. And so we've been looking very carefully at, the movements of these animals' arms and how does that affect locomotion. And it's really exciting to see the kinds of data we generate using this system. It's also daunting, but it's really opening up, wide swaths of science use cases, right? Being able to interrogate and look at organismal systems in such detail in their natural environment. And then lastly, I talked about live stream oceanographic. We've really been, getting more and more interactions and engagement with enthusiasts. And one of the problems that, Blair had mentioned, I know we all are very aware of, is the fact that we collect so much visual data just to do basic observations of life in the ocean. And we, on a large scale, like community-wide, don't have good solutions for processing all that information very, very quickly. Blair's techniques are pretty incredible. And then other techniques, though, require, identification down to genus or species level, right? IDs of animals. And so that isn't something that's very easy to do, nor is it scalable, the current ways we've been doing it. And so we've been standing up a program called Ocean Vision AI. FathomNet is this global labeled image database that anyone can use. But in talking with Livestream Oceanographic, what we're doing now is we're actually releasing a video game called Fathomverse that will, the minimum viable product goes live in July this year, and the full version of the video game goes live in January. So the idea is to have anyone participate in ocean exploration and discovery and help us identify life as we collect this information and contribute meaningfully to these data analysis pipelines. So Schmidt Ocean Institute is actually involved in Ocean Vision AI, so it's really exciting to see how that's going to go.
Yeah, that sounds like great fun. You'll have to share your video game, and maybe Blair will apply his algorithms to it and get lots of points and win. John, could you share what you've been up to, how you have been using the advanced technology? And I know you mentioned analyzing some of the DNA data earlier, and we actually had a question from an audience participant on whether that data was publicly available yet, and if so, where. So if you'd like to put a plug in for that.
Yeah, so I've had my head in a computer for the last year and almost 2 years, I guess now. It takes quite a lot of time to analyze that data still, because you're getting the entire picture of the information from any new organism. But we learned some really neat things already. So we got one of the smaller ones. It's called a SALP. We got its whole genome, and it's the most complete genome of SALP that anybody's ever sequenced. And it tells us a little bit of an interesting story. That's a picture of the genome. It tells us a story about some really interesting processes that are happening in it. Like the type of jelly that it's made out of is a little bit maybe surprising. There's a type of molecule in the matrix of our own bodies that they might be using, and SALPs are related to us in the animal world. So it's really interesting, although it's not a complete picture, because some of the things that should be there are missing, but some of the things that we see are so blowing us away that maybe there's something else, that maybe they do it in a new way. They use the sugar in a new way. So that's really cool. For the worm, the Tomopterid, the swimming worm, it has these two long processes coming out of its head almost, but they're actually modified tentacles, like grabbing tentacles. And it uses them kind of like tongues. So we're able to use this, the two long things coming out of the sides there. So it has actually real antenna that it uses for sensing, but they're really small. And then it has these two long modified tentacles that it uses to taste the water. So they're actually like long tongues, and it might be sensing light. It's definitely sensing sugars and ammonia, and it hunts small fish. So ammonia, they excrete ammonia. So I think that, we get some really cool information from the genetics of these organisms. And that's what I've been studying. Yeah, that's a picture of me looking at the genetics. And, when you see these thick bars that all are the same color, it also tells the story of how well the techniques worked underwater, that we're able to get very consistent information from multiple samples.
So interesting. Thank you, John.
Yeah.
So we had a question.
The data, sorry, the data is
go ahead.
Not quite available yet, but as soon as we get that paper published, it will all be freely available on the national NCBI. It's called the National Database for all this information. So we're going to publish all the genetic information as complete assemblies as we can.
Excellent. And we'll also link to that data set from the Schmidt Ocean Institute webpage. So when you have the information, send it to us. And for our audience, keep checking the SOI website, and that data will be linked. We also had a question about where to read about all of your past projects. So I just wanted to remind our participants that they can check the Schmidt Ocean Institute website. We had a question about when you were coming back out on Falkor 2 to test or bring some new technology. So I will remind you guys and also our participants that Schmidt Ocean Institute's call for expression of interest is now open for science on Falkor 2. But to tie that in with another question that we got, and this might be our last question, but someone asked if there were interesting scientific targets, either biological or physical, that lack effective robotic sampling techniques. So are there places in the ocean, either biological or physical or chemical, that we don't yet have good technology for sampling and understanding the ocean? And when you think of your answer, feel free to submit an expression of interest to come use some technology and answer that question on board Falkor 2. So if anyone has a response to some knowledge gaps in the ocean that we still need to develop techniques for, we would love to hear them. Blair?
Yeah, so I guess one question is, where does the ocean stop being the ocean? So when you go to the seafloor, there's fluids inside the sediments, there's fluids inside, the Earth's crust. So once you start kind of crossing that boundary to under the seafloor, I think that's somewhere where a lot of the robotic technologies we have, we can go out there with some acrylic tubes that we can push about 30 centimeters underground, but that's about as far as we can go with the robotic technology. So I think that's a very exciting area, is kind of what do you do across that interface? And I'd say the other challenging area is the other interface, the one at the top that we paddle around and swim in. So there's a lot of stuff that happens across the atmosphere ocean boundary. And that's also something that's pretty difficult to study and observe. But I think that idea of kind of interfaces across boundaries is challenging, because we build things that are good in one of those mediums, but not multiple mediums.
Sounds like a great expression of interest.
I have something to add to that. I think where should I start? So, for instance, right, the ocean is massive. How do we do global scale observations in the ocean? Not just at the surface, right? Or not just at the seafloor, but the whole volume, right? The ocean is 97% of the habitable ecosystem on our planet. Depending on who you talk to, we've maybe, everybody talks about mapping the seafloor. I think we've reached 25%, the same resolution as the moon and Mars. But again, the ocean is a massive volume. So what is that? We may have explored about 1% of it with imaging or with our own eyes. So I think, yes, there's still a lot of challenges or places that, robots still can't observe, but then there's also the scaling problem. How do we actually scale our observations? And, we need to do that if we want to not only meaningfully make a single observation, but also actually monitor the ocean, which requires repeat observations. So, in my mind, that's one of the biggest challenges. And how do we do that?
Definitely. John or David or Joao, do you have anything to add? Yeah, David?
I would like to think of, one of the things to think about when we're studying and we're using this remotely operated vehicle with like big thrusters and huge lights. So really the things that we see are the things that it might just be a fraction of the things that are there. So I think we could even think of going there as quietly as possible. We've been exploring low light technologies and sometimes even using just a little pen light to drive the ROV with sophisticated cameras. So, I always think whenever we do even biodiversity studies of coral reefs, you might see a few species to the eye, but then there could be, many orders of magnitude more that are there that you're just not seeing. So how to be more surreptitious, how to be more invisible in the background is something. And I think I also just wanted to point out, because I was kind of watching over John's shoulder when he was doing the analysis, and just how hard it was, because most of this genomic technology is developed for humans and not really for creatures of the deep sea. So even looking at the quality of the DNA, that the indices that we use are not really meant for these animals. So there was so much exploring and knowing how good of the quality the genomic sample was. And it was really this windy road to try to even come up with what we came up with from this. So yeah, just to kind of highlight how exploratory this is from, even from the biological side in thinking of the techniques.
Great, thanks. John, do you have anything to add to that?
Not even on the genetics. One thing about the scaling the oceans is I think there's cultural aspects too. So one of the west coast of USA is probably one of the best explored regions of the ocean's midwater, but other places that don't have the same resources are totally unexplored, and our ROVs can access them, but there's reasons that they're just, we don't get to them, or the countries that have them in their borders don't explore them right now. And I think that's a big challenge too, to make all this accessible to the world.
Yeah, that's a great message, John, not just developing the technology, but bringing it to new places or to countries that don't have those capabilities on their own. Joao, do you have anything to add before we wrap up?
Yes, just a kind of a small comment about temporal and spatial resolution, which is, I think that we have to go several steps into that direction if you really want to understand the kind of smaller scales and what is going on those scales, because we are used to kind of a bigger scales, a kind of a mesoscale, submesoscale, and that type of stuff. And I think we still don't understand that well what's going on at the kind of smaller scales.
Yeah, that's an excellent point. And what happens at the smaller scales is typically what drives the larger scales and the biology of the region. So it's important to have that holistic view. Well, thank you so much, Blair, Kakani, Joao, John, and David. This was a really great conversation and look back at all of your expeditions that happened on board Falkor and the technology that was developed and used. And I'm really excited to hear what everyone has been working on, and excited to hear about the video games and new publications. So I know you have a lot going on. I wanted to thank you for taking your time to sit with me and share all of that information. I wanted to remind our audience participants that Falkor 2 will be back at sea conducting research on June 2nd off the coast of Costa Rica with Dr. Beth Orcutt and the Octopus Odyssey expedition. So look out for the live streams of that. And I hope to see everyone very soon. And thank you so much.